who may not have been able to attend today. If you know of someone that really wanted to be present this morning but weren't able, then please let them know that it will be recorded for them to view at a later date. Now, before we start, I thought I would do a short karakia to open up this gathering. And please, I apologize if my pronunciations are not 100%. E te hui, whai te mātauranga, kia mā rama, kia whai take ngā mahi katoa. Tu mai tu kaha, aroha atu, aroha mai. Tatui tatu katoa. And you can see the English translation underneath. For this gathering, seek knowledge for understanding, have a purpose in all that you do, stand tall, be strong. Let us show respect for each other. So first up this morning, I would like to introduce Dr. Emma Spotter. Emma is head of the New Zealand Motor Neuron Disease Research Lab that's based at the University of Auckland. After completing her PhD in the University of Auckland, Emma was awarded a Marie Curie International Incoming Fellowship in 2010 to go to King's College in London. She returned to the University of Auckland in early 2014 after being awarded an Aotearoa Fellowship by the Centre for Brain Research. Emma was awarded a Rutherford Discovery Fellowship in 2016 to support her vision of growing a national motor neuron disease research programme, and with her team established the New Zealand Motor Neuron Disease Research Network in 2017 with funding from Motor Neuron Disease New Zealand. Welcome, Emma.
I'll just unmute myself. Thank you, Dimpna, for a lovely welcome, a lovely karakia, and thank you everyone for coming along today. This is the second in our webinar series, so some of you might have joined us last time, but it's my pleasure today to be able to talk a little bit about the work that my lab does. So I'm just going to share my screen. If anyone has any problems with audio, with hearing me, please just unmute yourselves and make a noise, or you can raise your hand virtually. Okay, those slides okay for everyone? I get a thumbs up. Great. Okay, so as I said, it's my pleasure to introduce the work that we've been doing over the last couple of years, really, with a few interruptions rather from COVID. And I'm going to talk mainly about the motor neuron disease genetic study that we're running across New Zealand, but I'm going to bring in some little hints of the other type of work that we do, including the pathology, the neuropathology, so looking at brain tissue from people who have passed away from motor neuron disease, and also examining some of the biological mechanisms of disease more in real time. So we have cells that we grow in a dish, and we are able to look at biology as it's happening rather than capturing things at the end of life in tissue or looking at genes without examining their products in living people. So I've got this slightly out of date image of our lab, but there are around 10 of us. Have a postdoctoral fellow, Molly Swanson, 5 PhD students, some honours students, obviously Dimpna is part of the team, and together we are working on overlapping but separate questions about motor neuron disease, mostly about causation, but also how we can circumvent or redirect the course of disease, whether that's in cells, whether that's looking at what happens in brain tissue. So this is my team during one of our lockdowns, and we continued our work mostly online and through Zoom, but we're very pleased to be back in the lab. So really feeling for Luke, our other speaker who's locked down currently. So as I said today, I'm mainly going to be talking about our genetic study. So I've got that title a little bit larger, and I'll just touch a little bit on our brain tissue studies and our cell biology studies. So I really don't need to introduce motor neuron disease to this audience, I'm sure. But what I briefly wanted to say is that some of our understanding of motor neuron disease, some of the kind of tropes or dogmas of disease are really changing based on current research. So we understand that around 5 to 10% of people with motor neuron disease, and actually I'm probably going to use the term ALS. I tend to use a mixture of these terms throughout most of my presentations. In the United States, ALS is the more common terminology, but in New Zealand we use the more overarching term of motor neuron disease, where motor neuron disease actually encompasses both ALS, that's the most common form, but also the three other forms, progressive bulbar palsy, progressive muscular atrophy, and primary lateral sclerosis. So those are less common, and they affect either the upper or lower motor neurons selectively, whereas ALS affects both. So I'm going to talk about ALS as a kind of catch-all term, and what I mean is ALS or MND. So we see around 5 or 10% of people who present with ALS will have a strong family history. There are multiple people in the family who are affected by disease, and those people are closely related. But if you look in those families, the number of genes that you can find are quite extensive. So there are around 40 genes that are known to cause motor neuron disease in families. But we know that those gene mutations are not the only factor, even in those families. We could talk a little bit about that on the next slide. But basically, I'm going to explain a little bit about the multi-step hypothesis of motor neuron disease, that there's this interaction between environmental factors that people are exposed to over their lifetimes and the genetic hand that they're dealt at birth. And through mathematical modelling, Amar Al-Chalabi and his group at King's College London has shown that there are 6 individual insults or steps that need to accumulate over a person's lifetime for them to reach a threshold where they manifest motor neuron disease. And here I talked about the difference in the motor neuron diseases affecting either these upper motor neurons with their cell body situated in the motor cortex and projecting down either to the upper or lower spinal cord, depending on whether they innervate the upper or lower limbs. And for some people, only one set of these 2 neuron groups will be affected. But for ALS, the most common MND, it's both sets of neurons. So this is the working hypothesis that our group subscribes to, that was described by Amar Al-Chalabi, that if you look along this time axis here, many people who present with, I'm going to say sporadic MND with inverted commas, many people will start their lives with no predisposition to motor neuron disease, but then over time, there will be a series of steps where molecular damage, and this is where a lot of the question marks are in motor neuron disease research. What are the molecular damage factors that can accumulate or can interact to cause MND? And these are some of the molecular damage factors or environmental factors that there is better evidence for. But even with these, the relationship between these exposures and motor neuron disease is much less certain than genetic causes. And the reason is, if you think about all the things a person is exposed to in their lifetime and you try to work out which one, or even more difficult to do, which combination of factors can cause someone's MND, that search space, as Amar and another very good motor neuron disease clinician, Angio Chio, like to say, the search space for these factors is infinite. Anything you're exposed to in your day or in the last decade or in the last several decades could be contributing to the development of disease. Whereas when we think about genetics, that kind of search space for causes is finite. There's only a certain number of genes. So this is difficult to understand, but it doesn't mean that it's not possible. And if we consider people with sporadic disease, they are generally having to accumulate those 6 molecular damage factors before, in older age, they might reach the threshold for development of motor neuron disease and physical health declines. Whereas for some other people, they will be born with a higher predisposition. So what this tells us is that genetic factors that people are born with can actually contribute between 2 and 4 of these molecular damage steps, so that fewer steps are required to develop MND. There's much more certainty of developing MND, but again, not complete certainty of developing MND, because if these factors are not encountered during one's lifetime and this predisposition is not as high, some people don't develop disease. So really what we're thinking about now is, rather than sporadic MND and familial MND, we're thinking about a spectrum of disease where some people have really very only environmental factors that probably contribute to disease. Some people have strong genetic factors that still require some environmental contribution, but it's a real spectrum, and some genes have less effect, some have more effect. And we don't actually know, even at this point, whether it's true that there's no predisposition. It may be that even people with sporadic disease have weak genetic contributors. So Emma and Alice's group have said quite rightly that thinking about sporadic and familial MND as very separate entities and treating them and treating patients with sporadic and familial MND very differently is probably not the right approach. And our idea of whether someone has sporadic disease can be very strongly influenced by whether an accurate family history was obtained also. So it might be that a person with seemingly sporadic disease out of nowhere, when more digging is done, we might find things like a parent who had personality changing Alzheimer's, which might be frontotemporal dementia. And, again, that is on the spectrum with motor neuron disease. So really the family history, if not taking a good family history, can lead you to believe someone has sporadic disease. So I'm going to leave those in inverted commas when I talk about them, but I will say that if you do separate groups of people by who has a very strong family component to the disease, they have multiple affected family members, and people that don't seem to have affected family members, you actually find the same genetic causes in both groups. It's just that the proportions that they account for are different. So in familial ALS or MND, the most common gene that causes disease is C9orf72, and the same is true for people who have sporadic disease. It's just that this accounts for far fewer people. What is really important to consider, though, and this is a bit of a mathematical challenge for you all, is that this group of people, the absolute numbers in this group are really a lot larger than this group. Remember I said only 5 to 10% of people with MND might fall in this group, but all the rest have sporadic disease. So the absolute number of people in this tiny green bar is actually higher than the absolute number of people in this green bar here. So we're really interested to make sure that people who present with sporadic disease, who are the vast majority of people with MND, are able to access genetic screening and genetic therapies in the same way that currently in New Zealand, people with familial MND can do. So we designed our MND genetics study to do just that, to enroll both people with sporadic MND and familial MND. And we also enroll a small number of controls. We're interested in the genotype of these people, but mainly their skin tissue that we use to compare with skin tissue of some of the people in this group. And I've got these little approximately signs because this was our projected enrollment. We, based on kind of 5 to 10% of people having familial disease, and we wanted to enroll a total of 300 in this group, and we kind of did a bit of calculation. That's what we thought we would see in New Zealand. And the aim of the study is to have all of these people go to their local pathology lab in any city in New Zealand, wherever you can find a path lab, give blood, and that gets forwarded to Canterbury Health Laboratories, who are our partner in this study. They extract DNA from the blood, and then they send it to our other partners at the University of Queensland, who do the screening for us. But they also keep some of the DNA so that they can do retesting for us. And while the University of Queensland does what we call research grade screening, it's perfectly trustworthy, but it's not accredited testing that we can return information about to patients, whereas the testing done by Canterbury Health Laboratories is accredited, and we're able to return that information back to the participants in the study. So on the research genetics side at University of Queensland, they are screening for a panel of the 4 most common genetic causes, C9orf72, SOD1, TARD-BP, and FUS. And also this guy, UNC13A, which is a risk factor associated with motor neuron disease, but it can also be a modifier of the rate of progression of disease. So we're interested to see, some people might have a combination of this modifier gene and mutations in these causative genes, and that might alter the rate of progression of their disease. We also do some genome-wide screening, which helps us to understand what ancestral grouping people fall into. So we do the right kind of comparisons against controls. And we can also get some idea, we can infer some more genetic information about less common genetic causes here. Something we've just started doing that we're really excited about is some relatedness analysis. So this enables us to look at our New Zealand participants and compare them through our collaborators at Macquarie with people who also have MND who are based in Australia or the UK. And in that way, if we find very distant relatives, we don't use their names or anything, everything is coded, so we're not, there's no revelation of anyone's personal details, but we can identify coded individuals who are distantly related to our individuals in our study. And by looking at the common DNA elements between the related individuals, helps us again narrow that search space down for a causative gene. And the other thing we do is we look across the entire genome, every single base of the genome, looking for changes. And that is through Project Mine. And this is something we haven't started yet, but we've got our first set of DNAs that we're kind of packaging up and getting ready to send to Project Mine. Then if we find any causative gene changes through this research grade screening, we can go back to that part of the DNA that we kept in New Zealand in Canterbury Health Laboratories and ask them to do a test, an accredited test that then we can return back to the participant. And the return is done in a very sensitive way through the neurogenetics team, who are our collaborators. They're, Richard Roxburgh is the co-lead on this study. Miriam Rodriguez and Christina Buchanan do the consenting, and they return these results. And that's a really important part of the study, to tell people whether they have a gene change. And really importantly, that we didn't really appreciate how important this was at first, telling people that they don't have a gene change in any of the common genes that we've looked at. And that gives people a lot of, it relieves a lot of worry. And we're finding that some of that work is bearing out to be the most impactful, that affects the most people. So I said that we thought that we would recruit roughly these numbers of participants in each category. And this is what so that was what the projection looked like as a little pie. And this is where we're at the moment. We've so far enrolled 130 people. We're always looking to enroll more. So please, if you are a person with MND thinking about being involved in the study, or if you have people that you work with in your allied health profession or your research that would like to be involved, please put them onto us. We would love to enroll everyone that we can. And what you see here is we're roughly looking at the same proportions that we projected, but we have this extra category. So as we started the study, we realized that the families that we were working with, so people with familial MND, multiple members of the family are affected by MND. There were also members of their family who were not affected by MND, but who wanted to be part of the study. They wanted to help us identify the gene in their family, and they wanted to contribute their sample. So that group of people have been really amazing to work with, and those people are really helpful for us to try to find a gene for the family. This is what the breakdown looks like of the families that we're working with. We've got 14 different families where, sorry, I'm just going to move everyone's faces. Most of them have the C9orf72 mutation, which is what we would predict. It's the most common. A couple with SOD1 mutations, one with a ubiquilin-2 mutation, and 5 of these families we don't know the gene for. And so we're really excited to be able to try to find a genetic cause of disease in these families, help new members of the family or members who have not been tested to be tested for the correct gene, and give some more certainty to these families about who is at risk and who is not. So the way we've been managing the study has been using an online encrypted database called REDCap, and we're able to collect information about people so that we can contact them. We record all their recruitment and consent information. We record information about the coding and the numbers that we assign to their samples, and we also collect their results. So we have a very organized way of retaining information about everyone that's very safe, and it means that we can always refer to people and their samples by a number rather than ever using their names in any communication with any of our collaborators. And this also allows us to very easily see where we're at in terms of the study. So we know that this is the number of people that have consented. This is how many people we've collected DNA samples from. Actually, this number is higher. This is slightly out of date. We're now at 90, it'll show on the next slide, might be 94 or 96 samples that we have results for, and we've collected skin biopsies from 30 people. And for the skin biopsy side of things, I haven't really talked about this very much, but we focus this on our controls or people who have a family history of motor neuron disease. Sorry, just trying to, nice. Oh, there we go. Here we go. So these are the findings to date of the first 94 people that we've genotyped. So we found pathogenic mutations, so these are disease-causing mutations in 7 people, which are within the C9orf72 gene. So these are repeat expansion mutations. And what's really interesting to us is that 6 of these people were in families where a number of people come to us in the study, they've already been genotyped by clinical genetics, so they know their result and we can validate that result. But we also were able to identify a person who didn't know that they had a genetic cause of disease, and so they've been dealt with very sensitively. So this is a massive impact for the family, because that person had believed that because there was no apparent family history of motor neuron disease in their family, that they didn't have a genetic cause of disease. But regardless of that, any offspring would be at risk. So we were able to identify the gene in that person and to let them know what the risk was and what the options are for any offspring. And same with SOD1, we found one person with a sporadic form. Well, they presented as a sporadic case, but they had a disease-causing mutation in SOD1. So what we hope is that for these families, even though we're giving them life-changing and potentially very upsetting news, we're giving them tools and providing wraparound care to help them engage with clinical genetics. It gives them the information if there are people in the family considering whether they'll start their own family, they can make decisions based on that knowledge rather than not having the knowledge. We've also found a number of people with the UNC13A SNP, or single nucleotide polymorphism, that changes the rate of progression and can be a risk factor. But what's really interesting is we sometimes see that change in control individuals. So it goes to show that variation in this gene is not always associated with motor neuron disease development. And that is really where it's not black and white with genetics or motor neuron disease and of most diseases. A certain gene change in one person might have a different effect in another person, and we can not necessarily predict whether a gene change in one gene will necessarily result in disease development, and even if we think it will, then we can't say when. We also found some gene changes in these genes, but as I say, we don't know if these are just normal variation between people or if they might have contributed to the risk of those people developing motor neuron disease. What's been good for us is we're able to pick out the ones that we think could have caused disease, and we go to our collaborators at Canterbury Health Laboratories and say to them, can you do curation on this gene? Can you look at whether the evidence in the literature and from other patients suggests that this caused disease? And they do that, and they can come back to us and say, actually, no, that is quite a common change that we see in people that don't have motor neuron disease. That's just normal variation between people. And that means that if the New Zealand clinical labs encounter those same gene changes again, it's already been curated. So our research study has paid for the assessment of these gene changes, which benefits motor neuron disease genetics in New Zealand, even past the length of the study. We're basically building capacity in New Zealand for continued motor neuron disease genetic screening of people even beyond the lifetime of the study. And again, I say we're returning negative results to people, and this is really important, and it's really meaningful to families to know that it's very unlikely that there will be motor neuron disease in the next generation. So one of the things that we do when we find these, when we find genes that we're not really sure of whether they have caused motor neuron disease or not, or they can be causative, is we go in and we can test the effect of the gene change in cells. This is some work that I did at King's College London quite a long time ago now, but we were looking in a gene called profilin-1, which is part of the cell's cytoskeleton, or the shape of the cell, delivering things around the cell. And there was this change from one amino acid to another amino acid, a glutamic acid to a glycine, and we put it into cells, and we found that this is what profilin-1 looked like in a cell when it had its normal coding sequence. These were some other changes that we found in patients in that same gene. When we put those profilin-1 mutations into cells, you can see that profilin-1 formed these clumps. So it's really clear to us that having these changes to the amino acids of that protein are damaging. Whereas for the E117G, it looked very similar to the regular coding sequence of profilin-1. So the cell evidence was not very striking, but then Brad Smith, who was the first author on this paper, did a really nice analysis of all the published papers that have described people with motor neuron disease who have this EU117G change, and the odds ratio, we call it, so the likelihood of finding this gene change in people with motor neuron disease versus finding that in the control population is around 3 to 1. So it's about threefold more likely to find this change in people with motor neuron disease. So we think that this might be associated, and we're going to keep digging in on that one. So just to reiterate, the impacts that this study have been returning those negative results to people and alleviating fears around genetic risk, engaging with MND families and hopefully finding genes for families that don't have a genetic diagnosis, and for families that do, helping to connect them up with clinical genetics so that members of the family who are interested in being screened can do that. They can have full wraparound care and also be visible to both the research community and the clinical community when clinical trials for gene-based therapies begin. This was something that that Richard Roxburgh and Miriam Rodriguez, our clinical partners, told me about just yesterday. And it's the fact that running this genetic study has really changed the way neurologists are thinking about motor neuron disease, and they're seeing many more genetic tests being ordered for motor neuron disease through clinicians, which is very exciting. So it means that people are not necessarily having to come to us, to our research study, to get genetic testing. It means that the clinicians and neurologists who are seeing people are seeing the value in doing this genetic testing. They're ordering those tests themselves. And this is exactly what we want. We want a situation where in New Zealand, beyond the lifetime of our study, people with motor neuron disease are being screened for the right set of genes, and meaningful action is able to be taken if something is found and if something is not found. And then the final impact, which is really why the study was set up in the first place, is thinking about gene-based therapies. So when we started considering running this genetic study, it was because in other countries, people were talking about using antisense oligonucleotides, or little pieces of DNA that go in and find the mutant kind of copy of the gene that causes MND, and it's not the gene itself that lives in the nucleus, it's the copy of the gene called the RNA that goes out into the cytoplasm, and it's a bit like the blueprint for the cell to make a protein, but that blueprint is a photocopy of the DNA. So these gene-based therapies go in and destroy that photocopy. So more photocopies keep being made, but the little pieces of DNA keep going in and targeting those for destruction. So it means that the mutant protein is not made. And these therapies are being tested. They're looking good, some of them. And some of them we have less information for. It's not that they're not looking good. But the SOD1 gene therapy is looking particularly amazing. That's the one that's been in clinic and in testing for the longest amount of time. And we're really excited about the idea that our genetic study can be screening people and having people ready for either being enrolled in clinical trials to test these therapies or to be delivered these therapies once they're clinically approved and available. So having thought about, screening for genes in people living with motor neuron disease and the impacts that we're having, the other thing that we're really interested in doing is understanding whether at the end of life, people that have already died of motor neuron disease but have very generously bequeathed their brain tissue to us to study, whether we can tell from the brain tissue itself what the genetic cause of their disease was, or what the cause was. And one of the ways that we are doing this is by taking brain tissue, making little slices of the brain and applying antibodies to that, which help us to identify individual proteins. So I showed you for the profilin-1 gene, we put a mutant version of that into cells, and you saw a green cell, and sometimes it was clumpy. What we're looking for in the brain tissue is the same kind of thing. The signature of motor neuron disease in those motor neurons that connect your brain to your spinal cord and your spinal cord up to your muscles. In those cells, if you have motor neuron disease, you tend to have clumps of protein, specific proteins that form clumps. So we want to know, if you look in the brain tissue, can you not only tell that someone has motor neuron disease by looking for clumps, can you tell what kind of motor neuron disease they had? What's the cause? What's the genetic driver of their disease? So in 2013, there was a paper published, really great review by a group at UCSD, I think, and they summarized what's been found in the literature, saying that almost everyone with motor neuron disease regardless of all those genes that we've talked about, those 40 different genes, almost everyone, if you look in their brain tissue, they actually have the same clumps. So isn't that astounding that you have all these different kind of causes up the top that collapse or go through this common pathway, and there's this one protein, it's called TDP43, and that guy, rather than looking all smooth and whizzing around the cell as it should do, it forms clumps and it becomes quite immobile and sticky. So most people with motor neuron disease have that protein TDP43 forming clumps in the motor neurons. Some people who have genetic changes in the SOD1 gene or genetic changes in the FUS gene don't have that clump, they have a different kind of clump. But it seems like all of these people, if you just looked in their brain tissue, they would be pretty much indistinguishable. So is that really true? Could we look in brain tissue and try to distinguish these people in terms of different genes that cause TDP to clump? Okay, so looking in the literature, looking at what other people have found, it turns out that different genetic causes, so I'm showing you the genes that we've seen in one of these previous slides, the genes, the C9orf72 gene, which is the most common, it was found to cause clumps in proteins called dipeptide repeats. These don't actually exist in people without that gene change. So these are all really specific to people that have that gene change. We can see clumps of those in the brain. This is what happens when you stain the brain with different antibodies, different colors. You see these clumps. They shouldn't look like this. And the same is true for other genes. We're really interested in the ubiquitolin-2 gene, and when it has a mutation, again, clumps. And we can see that these clumps are labeled by some little kind of rubbish tag stickers saying, take this away. So actually, when you dig down, it's not just TDP43 that you see aggregating in human brain tissue in motor neuron disease. It's not just FUS and SOD1. There are also these dipeptide repeats. There are also a whole lot of rubbish tag stickers, these guys, and there are some others where we know the gene is mutant and causing disease in people, and it's probable that the protein that results from that gene is clumping. So we did a bit more maths, and we think actually if we consider the same kind of proportions that we had before for looking in a brain tissue, you'd see SOD1 clumps or you'd see FUS clumps if people had genetic changes in SOD1 or FUS. And then previously we thought that all of that remainder that's not SOD or FUS was indistinguishable, but actually it is distinguishable. So we're starting to be able to look in brain tissue and infer or guess at the genotype of someone that in their lifetime. We can also screen and check that that's true, but it's going to show us that this is really quite a heterogeneous disease. It's much more, there's much more difference going on between people, and some of that difference is needing to be accounted for when we think about treatments. So these are the things that we've been trying to do. Can we use neuropathology to quickly infer genotype? Which is to say quickly, I don't want to diminish the importance and the incredibly generous donation that people are giving us when they're giving us their brain. But sometimes this can help us to understand whether gene changes that we see are important or they're causative more quickly than doing things genetically. And also, if we can we use the neuropathology to distinguish between different genotypes? So I've just got a few examples of this, and then I'll draw you into our cell biology slide, just one to finish, and you can go on your way. So the first case that I'm going to talk about, we did not know the genotype of this person, and they, in their lifetime, they had sporadic motor neuron disease, so no clear family history. So we stained their brain tissue, and what we found was that they had aggregates of TDP43. So we knew that the genetic cause could not be a FUS gene mutation or a SOD gene mutation, because those people do not have aggregates of TDP43, they have aggregates of FUS or aggregates of SOD. But what they did have is the dipeptide repeats that are really classical and only seen if you have the C9orf72 gene mutation. So I'm going to talk you through this image here. We do a really cool kind of brain staining called multiplexed immunohistochemistry. So we're able to use not just 1 or 2 antibodies to detect proteins, we're able to put on 5 or 6 different antibodies and color them in different colors so we can discriminate between them. And we're also able to remove those and put on another round of different antibodies again. So we can start building up a picture of cell types and aggregate types and anything you can imagine we can look for in this same piece of brain. and locate it back to the same position. So the code here tells us what each color is. These green and purple clumps are the dipeptide repeats that are seen only when you have a mutant form of this gene. So here we go, the green ones are much more common in this person. The purple might overlay, or I'm finding it quite hard to see the purple myself. What we found in this case, which was really interesting, is they also had the TDP inclusions, and I've said, well, TDP aggregates. I've said that before, that if you don't have a FUS or a SOD gene mutation, you will have aggregates of TDP43, but it's just not clear why. So all those genes can collapse down to that common TDP43 clumping. So we did see that. And we also saw that the rubbish sticker protein is finding these dipeptide repeats and saying, hey, cells, take these away, these are rubbish. So we were able, in that case, to quickly infer the genotype by staining the brain. We knew that it was a C9orf72 case. Here's another one that we're really interested in, which I don't have an answer for, but this is case 2. And again, a person who had no family history, and we didn't know the genotype. So we stained for the dipeptide repeats, and none of those. So it wasn't, the disease was not caused by C9orf72 gene mutation. There were no aggregates of FUS or SOD, so we knew there were no gene mutations in those genes. There were no aggregates in one of the rubber sticker proteins, which we see those in these 2 gene changes. We did see TDP aggregates, so it falls in that 97% of cases, doesn't fall into FUS or SOD. But we did see one of those rubbish sticker proteins forming clumps in an area of the brain that we normally never see unless one of these other changes is occurring. So there's quite a unique and interesting pattern of brain staining in this case that we've never seen before, and it tells us that it's worth digging a bit closer into the genetics of this person. So we've got lots of sporadic MND cases in our brain bank, and we care about every single one of them. And we run this kind of testing to get more information about all of those. And when we have to, when we want to know, is this person more likely to have a genetic cause of their disease or an environmental cause? When we see something like this that's really unusual, it leads us to think that there might be a genetic cause that we can identify. So at the moment, we're undergoing whole genome sequencing on that case and trying to really dig down and find a gene that might explain this novel pattern. Then the last slide I'm going to show about our brain staining is this one is really interesting and really close to my heart, because we work with a family who are based across New Zealand and Australia. There are some members in all different parts of the world. It's quite a big family. And they were described by a group in Australia as having a ubiquitolin-2 gene change. So both of these people were from the family. And the gene change that the family had was a threonine to an isoleucine change. And this change has only really been seen in this family and one other family. And while it seems very clear that it probably caused their disease, the evidence that we had did not quite meet the threshold. We have very specific criteria for saying whether something is definitely disease-causing or not, whether a gene definitely caused disease. And this one didn't quite meet the threshold for that. So we thought we could add some additional information to help determine if this gene change in the family definitely did cause disease. So we worked with Tipu Sadiq's lab at Northwestern University, and they had also some families with ubiquitolin-2 gene changes. And the gene changes that they had in that gene met that threshold for saying, yes, that definitely caused motor neuron disease in that family. It's very clear that that gene change caused disease. And so we got some brain tissue from that family. We were able to see these clumps of ubiquitolin-2 in the hippocampus. It's the memory part of the brain. And we know that this is a very rare thing to see. We almost never see it in any other kind of case. There are some specific cases where we see it, but I won't go into that. So this is kind of a signature of having a gene change in that gene. So then when we stained our case from the New Zealand Brain Bank and a relative banked in the Victoria Brain Bank, and we saw the same kind of spots. They're much harder to see in this case, but you can, if you look carefully, see little green clumps of ubiquitolin-2, and same here, clumps of ubiquitolin-2. We knew that this was really good supporting evidence that this gene change was disease causing because it's causing clumps in that protein. So it gives us really good confidence that the family that we're working with from New Zealand and Australia have a mutation in their ubiquitolin-2 gene that does cause their motor neuron disease. There's not some other cause in the genome that's been not appreciated that was the cause of the disease. This is it. And that means that we are helping to contribute to evidence to kind of bump up the body of evidence saying that this gene change is pathogenic or disease causing. Once we're able to publish our study, we will probably move this verdict to likely pathogenic for that gene change. And the final thing I'm going to talk about is how we go back and look at the things we find genetically or in human brain tissue and try to understand dynamically some of the things that are occurring. And we can do that because we grow cells in a dish that we can kind of meddle with and make changes to in real time and see what happens. So one of the really interesting things that my PhD student, Charlotte Dunn, did was she stained a few different cells in a dish, and she stained them for that protein that we know forms clumps in almost everyone with motor neuron disease. And she found that if she had the cells under normal conditions, they're just treated with, it says vehicle here, we're treating them with the solution that this stressor is dissolved in. So it's basically has no effect. Then if we stress the cells, we see a really drastic change in our ability to detect the TDP43 protein. At first we thought it had totally gone from the cell. We thought the cell was degrading it or something else had happened to it, but we tested that very extensively. I won't go into that. And it turns out that this antibody that we're using is actually unable to detect TDP anymore once it's undergone this stress. So remember I talked about stresses being part of that molecular damage that happens during disease. So it could be that this chemical stressor that we're using here is modeling some of the chemical stresses that happen in people's lives who have motor neuron disease. It may be that people are being exposed to particular stresses that are changing the shape of their TDP43 and making it invisible to this antibody. So then we went back into the human brain to test that. Oh, sorry, this slide's looking a bit funny. So we went back to the human brain and we looked again with that antibody. This is the antibody that has lost the ability to find stressed TDP. But we have another antibody that can find TDP even if it's stressed. When we look in the human brain and we look at the clumps of TDP43, some of the clumps are found really well with antibody 2 and they're invisible to antibody 1. But there are other clumps that are found really well by antibody 1 and are visible basically to antibody 2. And there are lots of clumps that are just as visible to either antibody. So what we know is that because antibodies find things based on their shape, these are clumps of TDPs that are different shapes from one another. And we think we can use the shapes of TDP in people's brains to group them. And we're doing something quite interesting where we can basically plot how detectable a clump is with antibody 1 versus how detectable it is with antibody 2. We can group either the ones that are very antibody 1 heavy or very antibody 2 heavy and make a map of where those structures are and then go back to the brain and put the map back on. So we can find that TDP43, which we think might be in speckles, because it's these only found in these little dots in the nucleus. There's the map of it here. Those are recognized mainly by antibody 1. Nuclear TDP is recognized equally well, or in this case, equally poorly, by both antibodies. And we think stressed TDP43 is recognized only by, well, here it is. It's just in certain parts of this clump. So I'm going to come to a close now. Sorry, I've run a little bit long. But in summary, what we're doing in the lab is we are working with people who are living with motor neuron disease and trying to find either genes that are already known by the motor neuron disease community to cause MND, and we're also hunting for novel genes. We're using our staining of people who died from motor neuron disease, staining of their brains to understand the relationship between a certain gene change and the way motor neuron disease looks in a person. And we're also using the way the brain stains to help us look at those genes where we're not really sure if they cause disease. If we see that the gene change causes a clump in its protein product, then we can be a bit more confident that that might be disease causing. And then the final thing we're doing is looking at motor neuron disease in terms of cells, where we're able to manipulate live cells and to see what happens when we make changes. And that kind of models what might be happening during someone's lifetime, slightly picking the surface of that really complex and deep well of research that needs to be done about the environmental causes of motor neuron disease. So we obviously don't do this work alone. We have a number of collaborators in different parts of the world. We're very grateful to them. And I'm also extremely grateful to the really wonderful philanthropic and government support we have for our work. So thank you very much. I'm not sure if we have time for questions. I'm sure Dibna can tell us about time, but I'm happy to take any after or whenever suits.
It was wonderful. Thank you so much, Emma, for really explaining everything that's going on there, particularly in that genetic screening study. There was a comment from Beth Watson. She just wanted to say, Emma, this is exciting, both your actual research and the fact that clinical practice has been influenced. Thank you for your work.
Oh, thanks.
Really nice to hear.
So Beth was previously at Motor Neuron Disease New Zealand as the chair. What was your role, Beth? What's the actual name for what you were doing?
Yeah, I was the president.
President, yeah, sorry, not chair. Yeah, lovely to have you on the call, Beth. It's been wonderful to be able to do this work and seeing that the somewhat, what's the word? The slightly negative prognosis that people, well, obviously it's not a great diagnosis to have if you're being told that you have motor neuron disease, but the idea that nothing can be done and there was no point in investigating further, and that understanding the cause of your disease was not meaningful or helpful, I think that's really changing, and I think that gives people hope.
If anyone does have any questions, if you wouldn't mind just putting them into the chat, and then we can come to them towards the end. We're just going to take just a really short break, probably 5 minutes max, just to stretch, and then we'll come back with Luke to present. Thank you. Very short break, but I am delighted to welcome Dr. Luke McAlary, who is a research associate at the Illawarra Health and Medical Research Institute at the University of Wollongong. After completing his PhD under the supervision of Justin Yerber, Luke travelled to Vancouver in Canada to take a postdoctorate position with Stephen Plotkin and Neil Cashman. After 2 years, Luke returned to Australia and was awarded the Bill Gold Postdoctorate Fellowship to study motor neuron disease. Welcome, Luke, this morning.
Hello, just let me share my screen. Where is that button? Down here on this app. All right. So, right, good to go. So hello everyone, and thanks for inviting me on to talk. I really appreciate it, and I really enjoyed that talk by Emma just then. I thought it was great. Similar to Emma, or sort of a little bit different, in our lab here in Australia, we focus more on molecular causes of disease and understanding how they might be happening in a cell, and also how we might treat these causes of disease. And so I'm going to show you today one line of research we've been performing in our lab here, where we've been focusing on that SOD1, that SOD1 protein, and trying to figure out, are there ways we can prevent it from causing pathology? So that's what I'm going to talk about today. So I always like to go over the basics before I do any sort of talk. And this is one of the most basic and important things to note about biology, and it's the central dogma. Emma touched on this, but it's basically that DNA in your nucleus is transcribed into RNA, which is then translated into a protein. And this is what your cells are doing all the time, a lot of the time. And for those who are sort of more or less biologically inclined, we can think of it as though DNA is a library, the compendium of all information that makes you up. And that you can't, if you want to take information somewhere, well, you can't take the whole library, but you can take a book out of the library. And that's what RNA might be like. And then that book might have instructions telling you how to do something such as cook a hot dog, right? Now, unfortunately, like any form of text, we can have typos, right? So if you have a typo in your DNA or a mutation, this can translate through that pathway all the way to the protein. And these can be benign, or they can be deleterious and cause problems for you. So if there's a typo in the book in the library, it can lead to the creation of something not ideal, such as a burnt hot dog. So this is something that happens in biology and accounts for not all, but many diseases have mutations associated with them. And it's a good starting place for us researchers to sort of try and understand what's happening, right? So how might these mutations affect a protein, right? So our lab's very focused on the idea of protein folding, protein homeostasis. So this is the idea that once proteins are generated, they need to be at the right concentration in your cell, in the right place, and they need to be either degraded at the right time or created at the right time. And any imbalance in this might cause stress throughout the cell in different pathways. So typically, once your protein has been synthesized, it exists here in this unfolded state, and this is highly unstable. Proteins, or most proteins, might proceed to partially fold. So you can see there's little bits here that are a little bit folded, and there's unfolded parts, and this is sort of partially stable. And then the final result that we want typically is a protein that is fully stable and fully folded, right? Because stability prevents non-native or interactions that shouldn't be occurring within the cell between different proteins. So if a protein misfolds, it might form protein aggregates in the cell. And so Emma already showed us some clumping, and this is what we call in our lab protein aggregation. And so the thickness of the arrows here really tell us about how much protein might be proceeding down to aggregation from a certain step. So once a protein is fully folded, it's unlikely that it's going to aggregate again. It's more likely that proteins that are newly created are going to proceed down this pathway, although that is not a strict rule. And so one of the problems is, and we notice with motor neuron disease, is that these protein aggregates are associated with many diseases, in particular these neurological disorders that we see. So things like Alzheimer's, Parkinson's, Huntington's, prion diseases. And in each case, but not all, there seems to be specific proteins associated with each disease. So in Alzheimer's disease, we have amyloid beta and tau. Parkinson's disease, we have alpha-synuclein. In Huntington's, we have Huntington protein. Prion diseases, we have prion protein. And in motor neuron disease, we have the bulk of cases made up by TDP43. But of course, there is a little bit of overlap between these that's becoming more apparent as research is enhanced. Right. So one of the main proteins our lab focuses on is superoxide dismutase 1. This is for several reasons. It's incredibly well studied in the field of ALS. It was the first known genetic cause of ALS MND. And so we know a lot about it. And it also happens to be a protein or gene that, when mutated, results in a pure ALS phenotype. There is no known overlap between someone developing a SOD1 mutation and also developing frontotemporal dementia, which is something that Emma also touched on, which is a spectrum, a disorder on the spectrum of ALS. So it's pure MND. And we think that by focusing on this gene, we can figure out things purely about MND. What is MND at its core? So this protein, which I've got shown here as a crystal structure, it's an antioxidant protein. So it floats around in your cell cytoplasm and it scavenges up oxygen radicals that are produced. So a motor neuron can produce a huge amount of oxygen radicals. So it's obviously an important protein to have active in a motor neuron. It does this through the fact that it contains a copper ion. So this is this little atom here in blue, and this is the active component of the protein. It does contain a zinc as well when it's fully folded, but the zinc is more there to provide structural stability to the protein. And perhaps one of the most interesting things about this protein is that it can have a mutation almost anywhere in this structure, and that there are now over 160 possible different types of mutation that are associated with MND. So it seems that the mutations are particularly effective at destabilizing this structure. And it's apparent now that we know that these mutations do destabilize this protein. And I'll go through a little bit how. But if we come back to our idea of how proteins fold in a simplistic manner, this is basically what SOD would look like when it's fresh synthesized. So we've got two empty spots for the metals, zinc and copper, and we've got this little two red lines. So those would be forming later on what's called a disulfide bond, which is a very strong type of bond that helps stabilize proteins. So the first step when SOD folds is the acquisition of zinc, there in yellow. The next step is the input of copper into the protein by a protein called the copper chaperone for SOD1. So this is a protein which has one job in your cell, and its job is to put copper inside SOD1. And then we get formation of the disulfide bond. Now, stability increases as we head along this pathway. So we want to move this protein as far along this pathway as we possibly can to prevent misfolding and aggregation, right? So again, thickness of arrows showing us where aggregation might be coming from. There might be an arrow here, but it would have to be incredibly small because this thing is really stable. Most of the proteins in your body are evolved to handle temperatures roughly around 37 degrees, a little bit more. Superoxide dismutase 1 can be boiled and it will remain active. And in fact, it's so stable that some researchers were able to take a piece of tissue from a mummy in Egypt and purify SOD1 out of it, and it was still working 3,000 years later. So it's a very stable protein once it's here on this right side of the page. Now, if you've got a mutation that proceeds along here, we lose the protein reaching these final, more mature states, and more of the protein will proceed down these pathways, form aggregates. So this is bad. We don't want this. So we have tried to identify through literature and collaboration with others, any sort of small molecule drug that might help different points in this pathway, this network here that we've drawn. So one such drug, which is in clinical trials here in Australia right now, is called copper ATSM. And it's a drug that basically surrounds copper and through whatever mechanism that's still being worked on is really capable of delivering a lot of copper to the central nervous system. So this can help promote copper input into SOD. And then something that we worked on the first paper that looked at this is that there's a compound called epsilon, which can help promote the formation of this disulfide bond in the protein. So here we've got two molecules that potentially show stabilizing effects for the protein. And we want to look at these, right, in more detail. And this is what we think we want to get. Now, obviously, the network still has some deficiencies. There's still this species here proceeding, this here, and these existing, but anything that can help, because your cell can also help you with taking care of aggregates. It doesn't have to only be up to the drug. Alleviating part of the problem can help a lot. So to do this sort of research in our lab, we typically use these cellular models that Emma mentioned before too. So how we do that is we have our DNA mutation on it, and then we splice that with more DNA that encodes a new protein called a fusion protein. So here we have our SOD1, and it's fused to a fluorescent protein called GFP, green fluorescent protein. And this gives us contrast so that we can look at it with a microscope and see where it is and what it's doing in the cell. So if a cell is taking up the DNA we put into it, it'll glow green. So that's what a normal cell looks like, right? So this is wild type SOD1 in a cell. It's nicely, fairly evenly spread throughout the cell. There's some in the nucleus and there's a lot in the cytoplasm. But if we have a MND mutant in our cell, we end up with this clumping that Emma also mentioned. And so this is what we try to analyze a lot of the time. And we want to see, can we decrease the amount of clumping in these cells as a marker of any drugs we're testing working. So what we do and what we've sort of really developed over the last few years is this idea of drug screening in a sort of non-biased and high throughput format. So as much sample as we can get through, we want to put through. And so this is work that was sort of led mostly by my honours student, Victoria. And the basic gist of it is that we have many plates of cells, and we can take tens of thousands of images, and we look at the total population of cells using this DAPI stain that stains the cell nucleus. And then we look at those that were transfected or took up our DNA to produce mutant or normal SOD1. And then we can perform these sort of image derivatizations. You don't have to worry about that too much, but we can identify every single individual cell, and then we can measure every single individual cell's properties. And so this creates what we call a cytoprofile. And by doing this, we have basically a huge amount of information on an individual cell. We've got its size, how bright it was, how dim it was, if it was granular or smooth. And then we can basically take this population where we don't really know what each cell has inside it unless we look with our eye, and we can train an artificial intelligence to learn what properties make a cell have inclusions or aggregates and what properties don't. And so this lets us basically use a tiny little data set of these cells from our thousands of images that contain millions of cells. And we can basically look at over 1 million cells and say how many had inclusions and how many didn't. And so we've sort of set this up right now, and it's working very well. The entire lab now uses it pretty much because it's so effective. Yep. So not exactly from this, but from previous studies. So we had Natalie and Madison working on the initial idea of copper ATSM. And this came from trying to understand a more personalized idea of what copper ATSM might be doing, because it was and still is in clinical trials in Australia, and understanding, does the drug work like we think it does? And so this graph sort of shows basically on this y-axis the number of cells with aggregates inside it, and then we've got mutants down here. Dark gray is no copper ATSM, so that's nothing, just vehicle. And then light gray is with the copper drug. And so what you can see is that we've got this sort of almost across the board addition of copper ATSM decreases the prevalence of these inclusions in the cells. However, I want to draw your attention to these 3 mutants, because this is something that we hypothesized and ended up being true. And this is that copper ATSM will be less effective or not effective at all if a SOD1 mutation ablates the ability of the protein to bind the copper. And this seems to be the case because this G85R mutant is a very weak copper binding mutant. H46R doesn't bind copper at all. And G127X can maybe bind copper, but that X mutation, that's an insertion that deletes about 20% of the end of SOD1. So you've only got 127 amino acids. That's not going to fold properly. It's a poor folder, highly aggregation prone, and it doesn't seem copper ATSM is that effective, as it is in the case of these other mutants where we know copper can bind quite well to these mutants. So that sort of tells us, well, copper ATSM, great clinical trials, but we might need to formulate something else for those people who might have a different mutation if they have SOD1. And this really hits home the idea of personalized medicine and understanding the underlying cause of someone's disease, not just diagnosing them with ALS, but like Emma said, doing the genetic testing and understanding what their profile might be like. Right. Oops. Right, good. Okay, so, and then this is where Victoria followed up, so just epsilon. So we previously showed in another paper that epsilon can help promote the disulfide formation, but there was no, we made no effort at that time to look at decreases in inclusion formation, because we think that's the best marker to use for a drug that could possibly work here. So we've taken, again, several mutants, several variants of SOD1 in our cells. We're looking at the number of cells or the percentage of cells with inclusions, and then we're looking at a dose response of epsilon. So is there a dose response that can tell you if a drug's working or not? So here we can see this wild type doesn't form very many inclusions. Awesome. And then we've got variable inclusion formation for each mutant that we look at. That makes sense. Each mutant will destabilize the protein to a different degree. But really nicely, what we see is that, yes, we do see that there is a dose response of this epsilon compound in decreasing the number of inclusions that these cells are making. And again, this does not work for G127X. And that's because G127X doesn't contain one of the parts of the protein that's necessary for this disulfide to form. And that sort of helps us understand, yes, it's probably working through this disulfide formation mechanism. We also have other data that proves this, but I'm not showing it here today. We've measured the percentage of SOD that's disulfide bonded before and after epsilon treatment. Right, so Victoria did very well here with this sort of study. And so then we followed it up and we wanted to look at, is there some synergy between these two compounds? It's very legacy thinking and something that I'm trying to convince more people of in the neurological fields, that this one drug will fix one disease idea is going to work. It's definitely not going to work. Cancer researchers have moved far beyond this to multi-drug combinations. Antiviral therapies are multi-drug combinations. Yet in the neurodegenerative disease area, we continually run clinical trials that look at one drug on one disease when we know from genetic studies, transcriptomic studies, that the profile of patients is so different, and that the underlying causes of disease are so different, that although the end result is MND, there are many different pathways that go there. There are many roads that lead to Rome, really. So what we did is we said, okay, can we enhance both drugs by mixing them together? And so what you're looking at here are heat maps of each treatment. So each little box is a different treatment. And then each major grid is a different mutant. So here we have wild type, A4B, G85R, G37R, G93A, and B148G. We've got our scale here. So this heat map scale, the more blue the color, the darker the color, the less inclusions. So wild type, almost none, right? Very hard to see inclusions for wild type. However, we see for each one that from right to left, epsilon is increasing in concentration, and from bottom to top, copper ATSM is increasing in concentration. So we get this sort of decrease of inclusions as we trend towards both drugs being at their highest concentration. And so we found that basically we could decrease the concentration of both drugs by around 8-fold and still get incredible reduction in inclusions. And so this is good, because if people are being dosed with just one compound, they might need a lot of it, but if we can add something in, we can lower the dosage of both things, perhaps, and really avoid unwanted side effects, perhaps. So this is something we think is very good for patients. But unfortunately, G85R here doesn't respond to copper ATSM even when epsilon's present, and it only moderately responds to epsilon, and this is because this mutant is so destabilized. So again, obviously other compounds, other drugs might have to come into work here for us. But this is looking very good and we're very happy with these results. Right, so future prospects and things we're currently looking at are obviously these other pathways. So one in particular is this one, and even though I've got question marks there, we do have certain ideas that have popped up recently. So obviously we want to deliver zinc. However, delivering metals is often quite hard. You need some sort of chelating ion or something. However, reading about antivirals that are being examined for COVID, stumbled across a compound called quercetin that is a zinc ionophore and can carry zinc into cells. So that's something we're going to test once we're probably out of lockdown. And then we also want to order in a anti-cancer agent that is designed to deliver zinc into cells for reactivating anti-cancer genes in your cell. Obviously, we're still thinking about this. This component is likely to be a gene therapy at some point. It is hard to remove aggregates with a drug. There are some drugs that can promote your own intracellular protein defenses, and we might test those too and see if we can't really knock down this entire pathway and promote this healthy line of protein folding here. So I just want to summarize. We know that motor neuron disease is associated with protein aggregation. Doesn't mean the protein aggregates are the cause, but they're a good thing to attack because we seem to look, when they form inside a cell, the cells die most of the time. We think that preventing this aggregation or promoting proper folding may slow disease progress. We think that copper ATSM and epsilon in combination is something that we definitely want to follow up and move into animals whilst we work on other components in this formulation. And we're not going to stop. So we're going to start a unbiased drug screen once we're out of lockdown and hopefully find some new compounds as well in this model. And there's no reason this model can't be applied to things like TDP or FUS, right? The machine learning is unbiased and extremely quick and powerful. Right, and so I'd just like to thank the team. So this is the current team. So Justin for basically trusting me to run this sort of thing in his lab. Not many people would. Natalie and Madison, who worked on that initial copper ATSM work. Victoria, who worked with me on this machine learning setup and idea, which is really good. And then the rest of the team for just being great, and our funders. So thanks a lot, guys.
That's wonderful. Thank you so much, Luke, for sharing that great work that you're doing. Now we've got some time, so I'm going to open the floor to questions.
Hi, Luke. I put a question in the chat. So I know that a publication came out a few months ago saying that copper ATSM had shown benefit in phase 1 clinical trial. But the trial was run in people with sporadic ALS. Can you tell us a little bit about what you think the mechanism is there? Because you've described stabilization of folding of mutant forms of SOD1. What's happening in the sporadic ALS cases who don't have SOD1 mutations?
That's a good question. So I'll start that with the, I can only speculate, I don't know. There are schools of thought that misfolded SOD1 is associated with sporadic ALS, but not aggregated SOD1 in these cases, that the protein is merely misfolded but not forming large clumps. General knowledge surrounding proteostasis diseases is that it is actually the misfolded small forms of the protein that are the most toxic. But as whether this is true with misfolded SOD1, we don't know. I would say that copper ATSM is likely having several effects. I would think that a stressed motor neuron would require rebalancing of its metal homeostasis simply because under stress, more than just one pathway collapses, many pathways collapse. But I can't really do more than speculate, sorry. I think we need more research to figure this one out.
Yeah, thanks. It's very exciting though.
Right, is there time for me to ask a question?
Yep.
Hi, Luke, I'm Karen. Thank you. That was a really interesting presentation, and thank you, Emma. I really enjoyed yours as well. Luke, am I understanding correctly that you know, these trials that you're doing with the or you're looking at with the epsilon and the copper, that's in lab at the moment, and what's the path forward? What's your aspirations for the future of this research?
So the aspirations, and that's a good question too, where we're taking this, because we always think about the end result, like not just, you know, I'm interested in, you know, furthering science, but also there are patients that really need something. The aspiration is definitely to try and get these out. So the next steps include animal trials of this combination to see if they work in the SOD1 animal. So they're perfectly suited for the animal because the animals express SOD1. And then if they work in the animals, or there's some benefit, trying to get them into humans. But I also mentioned one of the other things, which was part of the aspiration is to try and change the thinking and try and spread that we don't really want to keep working on this one drug, one disease thing, because it's not working for us. We've tried so many different drugs, so many different treatments and therapies for all the neurodegenerative diseases in existence, and we haven't had a single good hit. So we need to start thinking about, you know, trying different methods and different combinations because nothing's working.
Any other? Oh yeah. Do you want us to respond to that, Kieran?
Sorry, I've just a few of the screens have frozen. Sorry, what was that?
Does that answer your question?
Yeah, that's really helpful. Thank you. And yeah, I found it really interesting about, you know, your comments about not one drug for one disease, and I guess that's where Emma's work is so important to really drill down on the nuances of this disease. So thank you very much for your work.
Thank you. Does anybody else have any questions? Still got a few more minutes.
I'll just make a comment. I think it will be interesting to see if some of the genetic therapies that are on the horizon, these antisense oligonucleotides or viral vector therapies, might be used in combination with the drug approaches that Luke's talking about. You know, just because something is effective, so the antisense oligonucleotides are looking pretty good, but there's no reason not to hope for an even better result by combination therapy. So I think the different modalities combining could be really interesting as well.
Okay.
I'd just like to say, Luke, sorry, I was just going to say, Luke, I'm sure lockdown really sucks for you, and I'm sorry for that, but it's been great for us to be able to hear from you via Zoom, so thank you.
Yeah, thank you.
Okay, I'll wrap up then if there's no further comments or questions to add. I just really, once again, would like to thank our speakers today. It's been fantastic. I'm just going to share, sorry, didn't mean to come up, but I'll put up. Yeah, it's been fantastic to hear all the great work that you're doing, and quite exciting as well in terms of what the future holds within the MND space. I'd also like to thank the Motor Neuron Disease New Zealand, who provide funding for the MND Research Network. And without their support, we will be unable to provide this information to you. We are hoping to have another networking event towards the end of the year. So more details will follow in the coming months around that. And in relation to the recording today, I'll be sending out an email to anyone that's on our mailing list, but I'll also be posting up it on our social media pages as well. So you can keep an eye out on Facebook and Twitter if you wanted to listen back to the recording again, or as I said earlier, if you know of somebody that will be interested in hearing what was said today, then please, I encourage you to share the information with them too. I hope that you've enjoyed today's speakers and the presentations. I certainly have. And I would just like to thank everybody for joining us on the call this morning. And I hope that you have a lovely day. Thank you, everyone.